All right, this section, I'm basically going to focus on the double crosses. I'm going to briefly mention pedigrees, but I have some other little videos in the next section on part 3 that goes through additional pedigree stuff for you. But like I said, I want to focus on the double crosses. So when we talk about dihybrid crosses, that means we want to know 2 traits at the same time. This is going to be a 4x4 table. If we have 2 characteristics, so here we're looking at, for example, we could be looking at whether they are axial or terminal, and on flower position or terminal, or round and versus wrinkled. All right. So when we do this, I always say we FOIL it. So we're looking at what are the possible combinations of gametes that we can have. Well, I could have big A and big R. I could have big A and little r. I could have little a and big R, and I could have little a and little r. So I have 4 possible gametes that I can have because they're going to independently assort. We talked about the law of independent assortment. And so when you put that in, so here we have our parentals, which were purebred. We get our hybrid, and then we're going to have our 4. Again, we're going to do our little, I call it foiling, if you ever remember your foil. And then you have your 4 different gametes and then your possible offspring. Now we look at the phenotype, we're going to have 1, 2, 3, 4, 5, 6, 7, 8, 9 that have the double dominant trait, yellow and round. So that gives us a 9. And then we have 1, 2, 3, round and green, 1, 2, 3, wrinkled and yellow. Those are our recombinants where they have 1 dominant trait and they have 1 recessive trait. And then we only have 1 down here that has both recessive traits. So our ratio is a 9-3-3-1. Our genotypic ratio would be something like 1 to 2 to 1 to 4 to 2 to 1 to, it's kind of crazy. But again, I can't see the genes when we're just looking at phenotypes when we're doing crosses. So this is our phenotype, and this is what you need to know for your lab. So again, because the results are supported by the assumption that all combinations of alleles of gametes were likely to mix and mingle, right, in the law of independent assortment, that is going to give us our different possible combinations for our gametes. Traditionally, when studying heredity in humans, we can then construct what's called a pedigree. A pedigree is a chart that we use to look at phenotypic issues. You know, when we look at whether they have a certain trait, maybe we want to know where did the red hair come from, or maybe we need to know where did the cystic fibrosis come from, a disorder. And so there are different types of disorders that we're going to talk about. But in this presentation, I just kind of want you to see what a pedigree looks like. And when we talk about pedigrees here, the squares are males, the circles are females. So what we're saying here is this is a couple. They had a son, and the son married a woman, and they had 3 children. The oldest is here, 1, the middle, and then the youngest. The youngest married and had 2 daughters. The oldest is married and had another daughter. So they have, we talk about our generations. We have generation 1, generation 2, generation 3, generation 4. Right. So we will talk about this more in lab. And like I said, I have some other videos that get more into detail on how to do these pedigrees. When they're shaded in, that means they have the characteristic or the disorder that we're looking for. If they're half shaded, that means that they're heterozygous, they're a carrier. If it's a recessive trait, We'll talk about the different types of inheritance patterns. We have what is called autosomal recessive versus autosomal dominant. And then we're going to talk about sex linked. So we're going to look at those in some further PowerPoint presentations. All right. So that is all I have for you in this particular lesson, and I will see you in the next video.